What do you think?
So fun.
I love my Christmas cards.
I did it with my mom.
It's very, I'm tired. This is for my boss.
Like I was sober and then you
I printed it. We need
like the
that.
lines and I.
I look at a picture online and this kind of like, I'm not creative, but I can still do this.
Very fair. I had pizza. We did it at my house. Because I'm not.
That kind of looks like your house.
Yeah, right. I was pretty sick.
That's right.
On last Wednesday, I was like, I'm hungry. Let me just go to the computer downstairs.
Oh, perfume. Okay. I was like, what is a cloud? Why does it want a cloud? I don't think this way. I think that people should know what I want based on my personality. I'm a very easy person to shoot for.
I'm a really bad.
I'm like, yeah, you're not doing the kind of like penetration. I don't know why. That's
Yeah.
like,
If you listen to this even little slightly, you will understand like my. Oh yeah, I do like thinking food, but like,
my sister's obsessed with
she watched it without me. No,
She's my mom.
so what?
We're seeing it on the left.
We are seeing it on the left.
I'm literally in love with it.
It's really sad that it came out when I'm selling mine.
Yeah.
Yeah.
I was in London when you died, and I was up in the theater district when you died. Yeah. I'm very upset right now. Literally everything that happened when I'm abroad anyway, so I can't go abroad anymore.
No, I have all of the receipts done. I don't have it in the
Hi, everybody. So lots to cover today. Just a reminder, tomorrow is not lab, but lecture time with me. Hooray. Thanks, I appreciate that. Okay. Please pander. Yeah, that's it for announcements. I haven't really started the grading yet, and so I'll get those midterms done and to you as soon as possible. All right, so let's get back into glycolysis. So as you recall, glycolysis has 10 steps, but we can break it down into two main components, the preparatory phase and the payoff phase. We're gonna walk through each of the steps of glycolysis. We're gonna start up here at the top in the preparatory phase where we're gonna take glucose and start making our first products. This is a chain of reactions. So every time you have a product of a given reaction, you shouldn't think of that as now floating off and disappearing into the rest of the cell, the cytosol of the cell, but really think about these as clusters of enzymes in these pathways that tend to be located together. And so these enzymes are almost passing their products on to the next enzyme as the substrates for their reactions. Okay, so these are all intrinsically linked. They're energetically linked because when we looked at the idea in the bioenergetics chapter of some compound X converting into some compound Y converting into some compound Z. If the X to Y transition was unfavorable, but the Y to Z transition was even more favorable, the overall X to Z transition was net favorable. So each of these reactions, they could be individually favorable or unfavorable, but in a network, they all contribute to the overall outcome of the products. And then we'll talk about how that whole system is regulated. All right, so step one, phosphorylation of glucose. As we discussed at the end of last class, this has a variety of different advantages. One of which is that the phosphorylation of glucose activates it is the, sort of the general term of art that's used, activates the glucose that allows for the energetic movement of that phosphate later on. It also locks the glucose in the cell, right? Once it comes across the plasma membrane, we have lowered the concentration of free glucose if we instead convert it into glucose 6-phosphate. And that glucose 6-phosphate does not go back out through the transporters. So now it's stuck in the cell just where you want it. And allows for that glucose 6-phosphate to go into a variety of different pathways. If you wanted to make some glycogen, you would start with glucose 6-phosphate. Yeah, so we are going to use ATP to phosphorylate glucose. And it's glucose 6-phosphate because we're adding it to carbon 6, right? Remember that one? So that's the one that's above the plane of the ring. The enzyme is called hexokinase. There's actually a variety of different enzymes out there that do the same type of reaction. Sometimes you might see it called, these types of enzymes called glucokinases. There are different hexokinases and glucokinases depending on species and depending on cell types. It's ATP dependent. ATP is a co-substrate, right? So both glucose and ATP have to bind in order for this reaction to occur, right? There's a cofactor, magnesium. Why do you need the magnesium? Anybody remember this? It's why we always include magnesium.
Isn't it stabilize the negative charge?
Perfect. That was the perfect answer, right? So we always include magnesium in biochemical standard free energy conditions, because if you don't have it, almost all ATP requiring reactions don't work. Magnesium as a divalent cation is like perfect for holding on and coordinating some of the negative charges of the ATP. So those positives and those negatives hang out, and it just basically makes the energetics of any sort of ATP phosphorylation hydrolysis reaction work better. 1 second. There we go. All right, so what we see here is under standard conditions, this is a favorable reaction. If you think back to the hydrolysis of ATP being generally favorable, right, there were 3 things that contributed to the overall big negative delta G of hydrolysis of ATP. Relief of charge repulsion within the molecule, the entropic gain of extra, like going from 1 molecule to 2, right? What's the third one? The extra stability of the inorganic phosphate, right? Because it actually has that resonance structure. Of those 3, which are we getting out of this reaction? Well, we count the numbers of molecules, right? ADP and glucose 6-phosphate, same number of molecules as ATP and glucose. So it's not the entropic gain, right? How about the resonance stability? No, there's no resonance stability because that guy's just stuck. And so we can't resonate with all the bonds, right? So what you are relieving though is that charge repulsion inside the ATP. So you're only getting a part of the whole benefit of that ATP hydrolysis. And actually that the rest of that benefit can come out later on in the whole process, okay? All right, so let's see, there's a couple of notes I have on here. Hexokinase found in eukaryotes, and there's an isozyme. Have you guys ever heard the term isozyme before? So it's a variant of a particular enzyme class, right? So so a particular function or location that it's like a subclass of the enzyme. And so in the liver, hexokinase, there's a version of hexokinase called glucokinase, not to be confused with the glucokinase that is expressed in prokaryotes. It all does the same thing, but does a little differently. Highly thermodynamically favorable, and thus we're going to consider it irreversible. It's obviously not. You had 1 molar concentrations of glucose 6-phosphate and ADP and 0 molar concentrations of glucose and ATP. What do you think would happen? You'd have a backwards reaction, right? That's just how it works. But under cellular conditions, it's fundamentally so slow, the backward reaction, that we can treat it as effectively irreversible. So I'm going to note that a few times, that sometimes things are so energetically favorable that the backward reaction is so small and slow compared to the forward reaction, but also consider that under cellular conditions, realistically, the backward reaction is just not occurring. Okay, so I'm going to note that in each of these slides as well. And there's a regulation note here that'll make a little bit more sense later on. But the enzyme, the hexokinase, is allosterically inhibited by glucose 6-phosphate itself. It's kind of interesting, right? The product of the enzymatic reaction will have an negative allosteric modulatory effect on the enzyme. Now, think about that as a regulatory strategy. What does that do? So as you create more and more product, you slow the enzyme down. So it slows the production of product. What that's doing is basically slowing this enzyme from just taking all the glucose and phosphorylating it, right? Eventually, if you have a lot of glucose 6-phosphate built up, that's because you don't have places for it to go. It's not continuing down glycolysis, it's not being added to glycogen, you just have a whole lot of glucose, right? And if you have a whole lot of glucose and you keep phosphorylating it, you're going to start using ATP. So you don't necessarily want to just like burn some ATP unless you actually need to make more glucose 6-phosphate. So as an energetic strategy, this makes a lot of sense. I have this clip of a paper here that shows that glucose 6-phosphate also has competitive inhibition of ATP binding, so it competitively inhibits one of the substrates as well as that allosteric action. So it's one of those mixed inhibitors that we learned about in the enzyme, right? So it's not just if it was just the allosteric effects, right? That would actually potentially be an uncompetitive inhibitor, but this is instead a mixed. And it has negative cooperativity. So there's a reason why you learn all this stuff. All right, so I think this is kind of neat to talk about the general hexokinase versus the liver glucokinase. This actually has health relevance. So you have glucokinase expressed primarily in the liver and also in the pancreatic beta cells. Anybody know what the pancreatic beta cells do?
Is that glucagon or beta?
That's the insulin secreting cells of the pancreas. When do you need to secrete insulin?
When you're hungry.
When your blood glucose is high. So it's basically the response after a meal, more or less. No matter what you eat, it's going to probably raise your blood glucose. Depends on what you eat. Like we probably all raise our blood glucose quite a lot over the last week. Okay? Starches will definitely do it. Booze and pie and all that stuff, right? So in response to increases in blood sugar, insulin is a hormone that's released by pancreatic beta cells. And what does that insulin do? Well, it signals to every other cell in your body, hey, there's lots of extra glucose, come grab it. And so it enhances the internal transport of glucose throughout the body. In the liver, it happens to really enhance glucose transport. Why the liver in particular?
Because that's where you have a lot of metabolism happening.
It's where a lot of metabolism does happen, but not the same way.
Alcohol.
Well, alcohol, yes, but the liver doesn't work the same way as the muscles. It's kind of often doing the opposite thing. What do we store in the liver?
Isn't it cholesterol?
Well, yeah, cholesterol, but think of glucose. Glycogen. Right, so the liver is going to be in particular sensitive to changes in insulin level, because if there's high levels of blood glucose, that's a good time for the liver to restore whatever glycogen storage it might have used up at some point, right? And so different parts of the body are going to respond differently to insulin levels. So what we see here are more or less the Michaelis-Menten curves of hexokinase 1, 2, and 3. Those are 3 different isozymes of hexokinase. They're in different tissues, they do different things, but they all kind of work the same way. You can see they all have a pretty rapid increase, a very far left shifted Km. And then the Vmaxes are all just normalized. That's why it just says 100% rather than a particular rate or something. So it's relative amount. So each of these HK1, HK2, HK3, like if you tested them in the lab, could have different Vmaxes per enzyme, but their Kms are pretty shifted to the left. So it takes a very small amount of glucose to get a lot of activity in phosphorylation of glucose. You can see the sigmoidal shape here for glucokinase in the liver, right? So this is not a classic Michaelis-Menten enzyme. This is showing that there's cooperativity, right? That's what that sigmoidal shape's telling you. And ultimately, the glucokinase in the liver is not inhibited by glucose 6-phosphate. Because if there's lots of sugar, you need to actually get it out of the blood and put it someplace safer, like all that glycogen in your liver. Because if it's elevated elsewhere for too long, it can actually cause problems. And this is where, when patients have diabetes, they cannot regulate their blood sugar properly. Too high or low if you have type 1, it's generally too high if you have type 2. And you're just basically not properly regulating how much blood glucose you have, and cells pay the price. So this is just one of those examples of all these little, annoying things I made you learn. Actually plays a role in just regulating blood glucose and making sure that you store glucose when and where you're supposed to. Oh no. I thought I froze. Yeah, I did. Why? All right, let's move on to the next slide. All right, so step 2, we made glucose 6-phosphate. Depending on what your cellular needs are, the glucose 6-phosphate will then convert into fructose 6-phosphate. All right, so glucose and fructose actually have a lot of structural similarity. They're basically, their orientations of their hydroxyls to the left and the right in your Fisher diagram are actually identical. Just whether you move the carbonyl from the end to carbon 2, right? Go from an aldose to a ketose. So in order for this to happen, there's an enzyme called phosphohexose isomerase, basically telling you exactly what it's doing, right? Grabbing a phosphohexose and isomerizing it into the other form. You can see this is a reversible reaction, requires magnesium because of that phosphate. This is a slightly unfavorable and thus reversible reaction, okay? And this is another thing that you're going to see over and over. Product concentration kept low to drive this reaction forward. Because one of the key things we talked about in bioenergetics is that the further you are from equilibrium, the more energy there is in moving towards equilibrium. Think of reactions like a spring, right? Not one that's like fully compressed or stretched all the time, but like one that has, gaps, right? If you push it down, it'll bounce back up, right? You stretch it out, it'll bounce back down. Reactions are just like that. If you consider pushing it down as you have an overabundance of products, right? It'll bounce back to equilibrium so that your products and reactants are now at balance. right? If you stretch it out, so you have too many reactants, it'll bounce back down until you have a balance of the product and reactants based on the energetics of the reaction. In this case, this guy is always going to be bouncing kind of up and down, like just around equilibrium, because it's going to be bookended on two ends, step 1 and step 3, by processes that are regulated. Okay, so it's just going to move as it needs to, producing fructose 6-phosphate when concentrations are relatively low, and bouncing fructose 6-phosphate back to glucose 6-phosphate if there happens to be an overabundance of the fructose. Okay. Here's a little bit of the reaction down at the bottom. I'm not testing on reaction mechanisms, but I know some of you more orgo inclined people like to see those sorts of things. All right. I don't know if this video works, but it's actually just a video mechanism of how the acid-base catalysis works. There's this link down here at the bottom, I think also shows the whole step-by-step process if you're curious. We'll move to step 3. So this is the second phosphorylation of what started as glucose. So we're going to take this glucose 6-phosphate, phosphorylate it again to make fructose 1,6-bisphosphate. Right? So again, this is one of those reactions that we treat as effectively irreversible, and it has a big negative delta G. You're using, you're hydrolyzing ATP, but again, this is one of those hydrolyzed ATP, but you're not getting the full benefit of the ATP hydrolysis, right? It's mostly the separation of the charges that matters. So now you have glucose 1,6-bisphosphate. So that means both of the carbons that are above the plane are now phosphorylated, right? Remember, because we converted this to fructose, so that means carbon 1 is now available. If you think about this, there's a couple of reasons why we did this weird second reaction. When I was in your seats a million years ago, right? I always thought, well, this is stupid. Why don't we just turn glucose into fructose? Who thought that up? And there's a couple of reasons for it. Well, one of them is if you look at the glucose 6-phosphate structure, this is actually kind of hard to stick a phosphate on because it's like right up on the ring, right? If you can bump it up here, now it's separated out. There's not this bulky ring in the way of phosphorylating that hydroxide. So that's reason 1. And then reason 2 is that ultimately what we'll see is that we can now kind of divide this molecule in half. And it's actually pretty close. It's very similar final products. That's what we're going to see later on. Okay. All right. So reaction 1, we already talked about how that's kind of regulated. It's regulated by the products, it's regulated by ATP levels, and a couple of other things. Reaction 3 is super duper regulated. There are lots of different mechanisms by which this enzyme phosphofructokinase 1 is inhibited and enhanced. Lots of things bind to it. So let's think about, what do you think are signals that would make sense to increase the activity of phosphofructokinase 1? How many of you watched the regulation video already? Yeah. Oh, oops. No, it's okay. Well, what I want is to kind of think about this logically, right? What is the overall final product that we really, really care about in glycolysis? Pyruvate and a net increase in ATP. Glycolysis is used to make pyruvate, but it's made to, it's useful, most useful for cells most of the time as a way to synthesize new ATP. So if you are an anaerobic organism, it is your primary, if not only, mechanism of making new ATP. So when is an important time to make new ATP?
You have a lot of ADP.
You have a lot of ADP and not a lot of ATP, right? So if you wanted to logically plan a way to regulate whether phosphoprotein kinase should be active or not, lots of ATP should inhibit it. Lots of ADP should enhance it, right? There are other ways that we can regulate it, but this overall idea of the final product in a chain of reactions activating or inhibiting some of the steps in between, that's called feedback regulation. If the final product is ATP and you've got lots of it and it binds the phosphoprotein kinase and slows it down, it's basically saying, don't bother with the rest of the reactions, we're good.
Do you think that PFK is the most important enzyme in this entire sequence?
No, most important?
Yeah.
No, because this whole thing doesn't work without all of them. Okay, it just happens to be the regulation is most important here.
Mhm.
It's most impactful. So if you have lots of ADP, suggesting you burned a bunch of ATP, right? That's going to enhance the activity here, speed it up. So you change your Vmax and your Km to basically enhance the production of the product. So this is a key regulatory step, right? Do not burn glucose if there's plenty of ATP. At this stage, you can go backwards if you need it.
Is there a way to directly phosphorylate fructose?
There are, in fact, actually, one of the slides earlier, yeah, here, right? So, there are ways to create fructose 6-phosphate directly from other mechanisms, from fructose, from mannose, from, and so there are ways, but the most common pathway is by, start with glucose.
You mentioned that the fructose, when it's phosphorylated, went down with a slightly different pathway, right?
Yeah, so fructose that gets taken up by the liver, which is where most of the fructose in your diet is going to go first, ends up in a fructolysis pathway that's slightly different and doesn't feed directly into this general glycolytic pathway. But fructose that ends up somewhere else in your body will probably just find its way into glycolysis. It's a weird thing. I don't quite fully understand it myself. It's like, why not use it? So this is basically what I'm talking about, right? Phosphoprotein kinase 1 is heavily regulated. ATP will inhibit the activity. AMP and ADP, right, monophosphate diphosphate, they're going to be signals that you used up a lot of ATP and they're going to enhance the reactivity. Citrate and fructose 2,6-bisphosphate also have effects. Citrate you're going to meet later. Citrate is a key part of the citric acid cycle, hence the name. Fructose 2,6-bisphosphate, we're not really going to talk about in this class, but it exists only in sort of specialized metabolic states, but it enhances the activity. Okay. All right. So the idea here is don't use up ATP in this step if you don't need to make some more. Right. All right, so now we've made glucose 1,6-bisphosphate, or sorry, fructose 1,6-bisphosphate. And we're going to do a classic aldol cleavage. We're going to break this into 2 bits. Okay. So we're going to take the 6 carbon sugar, create 2 3-carbon sugars, each individually phosphorylated. The 2 products are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. They're obviously different molecules. So the next step is going to be an interconversion of one into the other. And we'll talk about that in a little bit. Okay. The enzyme that catalyzes this is called aldolase. Gets its name because you're basically using an aldol condensation if you do the reverse reaction. Okay. If you take these 2 alcohols and form one larger molecule, right? This is a thermodynamically unfavorable and reversible reaction. So in order for this reaction to actually proceed at any perceptible level, you want your reaction quotient to heavily favor reactants over products, right? If you have really almost any products, you're going to grind to a halt. So if you keep your product's concentration low, this reaction will move in the net forward direction. Now, the next step, this also helps explain why did we bother converting to fructose in one step. So then the next step, I'll talk about that in a second. Let's talk about that. So this is a better visualization of why we converted into fructose. And it's not just so it's also that phosphorylation is easier if the carbon is above the plane of the ring, but it also makes this enzyme work. You have lots of different aldolase type enzymes, and evolution is good at taking an enzyme that's good at doing one particular thing, copying that gene, kind of letting it mutate into something else that does a similar type of reaction, but on a different substrate. So if you took an aldolase that was capable of taking glucose bisphosphate, 1,6-bisphosphate, and cutting in half, the mechanism that aldolases use actually relies on that carbonyl. And that carbonyl means that you can only cut between two carbons that are just a step away, right? So this carbonyl means you can cut here. This carbonyl means you can cut here. The mechanism of the reaction allows that to happen. So if you didn't convert to fructose, you couldn't actually do an even 3-to-3 split. You'd end up with a 2 and 4. The next reaction will convert the 2 3-carbon products into the same thing. We'll take one of them and convert them into the other. And then so you end up with a single product at the end of stage 5. Here, there's no easy way to go from a 2-carbon and a 4-carbon, sorry, 2-carbon and a 4-carbon, and you can end up with a unified product stream. And so this ends up making evolutionary strategy sense, because you can take something that exists in lots of different forms already in your genome for lots of different reasons, and modify it to work on fructose 1,6-bisphosphate, and voila. We'll skip the mechanism. So ultimately, this cleavage is going to result in dihydroxyacetone phosphate, glyceraldehyde 3-phosphate. Sometimes it'll be written as glyceraldehyde phosphate, sometimes glyceraldehyde 3-phosphate. Exact same thing. I put that down there in case different sources might confuse it. The glyceraldehyde 3-phosphate is already ready to go through step 6 in the process, right? But the dihydroxyacetone phosphate cannot. And what's key here is, if you look at this, if you wanted to convert these directly or down a stream of reactions to pyruvate, the ultimate product, right? You would actually need different sets of enzymes to do that, because these are structurally distinct. The type of enzyme that can bind to this and act on it are going to be different types of enzymes than this. So if you think about this as a broader evolutionary strategy again, do you want 2 separate pathways, or would you like a unified pathway? A unified pathway is ultimately going to be easier to control, right? You can regulate the system just using these one sets of enzymes, right? It means fewer genes and fewer protein products to worry about. So it ends up making sense to interconvert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, even though it's energetically unfavorable. So we'll just deal with it. We'll convert the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, but now we have a single stream for all of our products. So ultimately, we complete the preparatory phase by converting all of the products at this stage into glyceraldehyde 3-phosphate. This is also part of keeping the product concentrations low, right? So keep the glyceraldehyde 3-phosphate concentration low to pull this unfavorable reaction forward. I don't know what I did there. Okay. So these things combined give us a single stream of product. Glyceraldehyde 3-phosphate is going to be useful for step 6. It'll help make this most unfavorable reaction more favorable because you're actually going to reduce some of the product concentrations. And this is, I hope, slightly useful to sort of see where all the carbons in that initial glucose ended up, right? That initial glucose converted into fructose 1,6-bisphosphate, but the carbons numbered 1 through 6 are in the same order that you would see in glucose. Aldolase splits them up. You can see the structural differences here. And then ultimately you get to glyceraldehyde 3-phosphate, and the 2 phosphorylated ends makes sense. They were initially the 1 and 6, right? But the single unified molecule, right? Now, what we haven't done yet is really oxidize this, right? You haven't seen a redox reaction yet. And the way you know that, you haven't seen a redox pair. That's going to change. So once we get into step 6, you'll start seeing redox reactions, a redox reaction that's key to this process. And then we're going to end up making some ATP. So overall, what we did, we phosphorylated glucose, converted it into fructose 6-phosphate, phosphorylated fructose 6-phosphate again into fructose 1,6-bisphosphate, broke that into 2 bits, glyceraldehyde 3-phosphate, dihydroxyacetone phosphate, and then converted the dihydroxyacetone phosphate into more glyceraldehyde 3-phosphate. Overall, this process has 2 very favorable steps, which happen to be the most highly regulated steps, right? And a bunch of pretty unfavorable steps. So this is what we mean by we have actually invested some energy. Like, this is all standard conditions, of course, delta G's. So in the cell, it's a little bit different. But this gives you an idea of what we're working with. We did a couple of really favorable things, like one particularly unfavorable thing, one, meh, and then one kind of unfavorable thing, right? But all together, it's a net, basically, problem, plus 2 kilojoules per mole under standard conditions, right? So that means we are climbing up a hill a little bit. So ultimately, for all of this to work, we need to go down the hill in order for the payoff to occur. So we've used up 2 ATP, right? The net gain out of this process is going to be 2 ATP, right? So we'll get 4 later on. So you don't want to go through all of this unless you're going to have that payoff, which is why that 3rd step is highly regulated, right? Spend an ATP here, spend an ATP here. You're going to have a net loss of ATP for what? Some glyceraldehyde 3-phosphate? Okay. So what we'll do now is take those glyceraldehyde 3-phosphates, and keep in mind, there's 2 of them now. So we're going to do this in duplicate as we step through each of these reactions. Oxidation and phosphorylation, right? So this is actually where we're going to finally start to oxidize glucose. It's actually pretty far downstream now. And in the process, reduce NAD. Then we'll make some ATP, We'll do some reorganizations and actually end up making ATP in our last step as well as pyruvate. Okay. All right, so oxidation of glyceraldehyde 3-phosphate by GAPDH is glyceraldehyde 3-phosphate dehydrogenase. A clue: if you see dehydrogenase in the name of an enzyme, it's going to catalyze redox reactions. And if you're familiar at all with GAPDH, it's probably because you work in a lab that uses it as a a loading control in Western blots or in DNA, RNA analysis, so on. It is basically one of the more highly expressed genes and proteins you'll find in cells, and it's pretty much expressed at a very steady level because you always need to be going through glycolysis. You, in this reaction, take glyceraldehyde 3-phosphate and an inorganic phosphate that's just floating around, and you somehow manage to glue that to the enzyme, and it's not as negative or not as unfavorable as you think. You are, because you're pairing it with a favorable oxidation, the otherwise unfavorable addition of a phosphate to this molecule that already has a phosphate isn't so bad. What the subtle thing here is that NADH product may actually be worth more ATPs than the ATPs you're going to create. Okay. This is a thermodynamically unfavorable and thus reversible reaction. So it could go backwards if product concentrations are remotely high at all, right? So use the next reaction to pull everything forward. Right? Keep your product concentrations low and the reaction continues forward. So the product, 1,3-bisphosphoglycerate, oh, actually, yeah, I forgot that I added this. Looking at this particular reaction using some standard reduction potentials and so on. So we've seen NAD to NADH before, negative 0.32 volts and standard conditions. The oxidation of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate would be a plus 0.29. So you see the reduction of the bisphosphoglycerate to the glyceraldehyde 3-phosphate here. Right. So if you actually these numbers only go to 2 significant digits. If you actually evaluate this to 3, you get that'll give you this number here. So the electron acceptor here, right? Think about this reaction here. What is accepting the electron? What's getting reduced?
The NAD+.
Yes, the NAD+. And the trick for in biochemical reactions, you can almost always tell where the electrons are going by looking where the hydrogens are going. Right? Where are the protons also going? So you see NAD+ go to NADH, extra hydrogen. That's a clue, right? That's how you know it's more reduced. So it took the electrons, right? So that's the electron acceptor, minus 0.32 and 324. Okay. All right. The electron donor then must be the other, right? In this case, what's getting oxidized? Well, how can we look at this molecule and see whether it's oxidized or not? Right, so we converted glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. What's the structural change?
Losing a hydrogen and adding a phosphate.
Yeah, that's gone. We've added this. So that carbon lost control of one of its electrons, right? So it clearly got oxidized. So we find the redox potential of the reduction reaction here. We're going to subtract that from the electron donor, right, from the oxidation reaction. So minus the 0.291 volts. That gives us our voltage difference here, right? Then we plug that into delta G equals NF delta E and so on. That's how you get positive 6.3 kilojoules per mole. Hopefully that helps understand. Just to remind you, NAD, NADH, you're going to see it over and over. This is the start of just so much NAD, NADH for you. So NAD plus is always the oxidized form. NADH is always the reduced form. You'll see that closely related FAD, FADH, reduced FADH, oxidized FAD. There's a version of NAD that is phosphorylated called NADP. That's used in some other specific reactions. Same deal, oxidized NADP, reduced NADPH. So it's going to serve as an electron shuttle. And if you recall, I explained to you that the actual reduction of NAD is kind of unfavorable on its own. So it's a nice little storage of electron energy, essentially, that like ATP is for chemical energy. Like a little battery. Skip the reaction. And we'll go to step 7, where we're finally going to produce some ATP. Okay. This is what's known as substrate level phosphorylation. All the productions of phosphorylations that we'll see here are going to be substrate level, and what that refers to is taking an ADP and taking a phosphate from some other molecule and squishing them together, like stealing the phosphate from something else and making ATP. Okay, that's all the forms of ATP synthesis that aren't oxidative phosphorylation are substrate level phosphorylation. Oxidative phosphorylation is the one peculiar pathway that we will learn about at the very end of class. At the end of the electron transport chain, how do you make oxidative? How do you make ATP using the ATP synthase? This enzyme is called a kinase. What do kinases do? They phosphorylate things, right? We're phosphorylating ADP. So, more or less, every kinase is going to need some magnesium. We'll take our 1,3-bisphosphoglycerate and an ADP, produce a 3-phosphoglycerate and an ATP. And this is actually a favorable reaction. Kind of wild, right? We know how unfavorable it is to make new ATP, right? Because you're smashing the negative charges on in close proximity and so on, right? But in this case, if you look at the structure of this molecule, you have these phosphates in really close proximity, right? So it's actually similarly unfavorable, right? Additionally, look up here, right? Is there any reason to treat one of those oxygens different from the other? Is it really like that? The one on the left is definitely double bonded, and the one on the right definitely isn't. No, realistically, they would have a resonance stability, and that minus charge would be shared, right? And it's one of those strategies, right? If you end up with an unusually extra stable product, like this end here, you get rid of that phosphate, and suddenly this is a thing that's extra stable, right? That makes this work. Kind of gives you an idea where we might be going for the other one, right? This is a thermodynamically favorable reaction, right? Big negative delta G. But we consider it reversible because it's coupled to the GAP-DH reaction that we just discussed, which is unfavorable, right? And so between the two of them, right, if you consider what was the net delta G here, right? Plus 6.3, and this is super tightly coupled to that. And so now it's okay, now it's a minus 12.2, right? A little less exciting. And we'll see that there are other reactions coming up that are also, not all that exciting either. So because this reaction, even though it has a big negative delta G under standard conditions, actually exists in the middle of a bunch of reactions that aren't as big negative delta Gs, right? It actually exists kind of near equilibrium, or closer to equilibrium than you might expect in most cells. All right, so we're going to move that phosphate. see, it's on the end, right? That 3-phosphoglycerate, and ultimately, you can get a more high energy compound if you can move that phosphate off the carbon 2. This will make a little bit more sense, I think, when you see the, I think it's the next slide, right? I had some notes on here before, but anyways, the 2-phosphoglycerate ends up being more reactive in the next step because there's a difference in the reactivity between primary alcohols versus secondary versus tertiary, right? We're going to end up here with a nice primary alcohol on this end, whereas this secondary alcohol, not so easy to work with. So that's kind of the key deal. This is a weakly unfavorable reaction under standard conditions. So keep your reactant concentrations high, which is going to be useful, right? Because if you've got a big negative delta G right beforehand, right, you should be producing lots of products, which happens to be lots of reactants for this one. So that'll drive this forward, is because it's kind of like a waterfall right upstream, pushing this one forward. The strategy of the last reaction required that primary alcohol in order for this to occur, right? So what we're if you didn't actually move the phosphate, this reaction is much more difficult to catalyze, and then this condensation reaction actually ends up setting you up for a really big kind of final payoff. Okay, you'll see in a minute why phosphoenol pyruvate ends up being a great final product for this entire reaction. So you have invested a little energy here. We got a positive delta G, because if you think about what we're doing here, is we're actually just making this carbon even more energetic, essentially. And it's attached to a phosphate, and so it ends up being a little bit of an uphill climb. So we end up with a slightly thermodynamically unfavorable and reversible reaction. But if you keep your product concentrations low, it'll drive forward. In this case, the 2-phosphoglycerate that was your substrate is not a good enough phosphate donor. Right, that last step needs to move that phosphate to ADP. So you need that last step, just like we saw in the previous one. You want your other products to be unusually stable, right? The more unusually stable the products are, the bigger the negative delta G that you can get. And since we know phosphorylation of ADP is unfavorable, you need to get as low as you possibly can in terms of your energy state for the final products here. The final product of 2-phosphoglycerate is pretty boring if you just dephosphorylate it, right? It won't have any extra stability over what it basically already has. But phosphoenol pyruvate will have extra stability. So here's our last step, and this is the last production of ATP. Just to remind you, like in the previous production of ATP and in this one, we only see one ATP, but you have to consider we're doing it twice per molecule of glucose, right? Because we have 2 glyceraldehyde 3-phosphates that we produce, right? This is another phosphate, substrate level phosphorylation, requires magnesium, enzyme complex called pyruvate kinase, right? It's phosphorylating stuff, right? And our final products, ATP and pyruvate, we know that making new ATP was energetically costly, but because pyruvate is so stable, you end up with a big negative delta G. if the last step in a whole enzymatic pathway is like a big negative delta G, right? It's kind of like the last part of a river or something ending in a waterfall, pulling all the rest of the water upstream over the edge. So this is highly thermodynamically favorable, and we consider this effectively irreversible in cellular conditions. Highly regulated as well. This is, this is regulated by ATP, a bunch of divalent metals, other metabolites in this whole process, because this is basically one of those irreversible steps. Generally speaking, there's a strategy. Things that are going to be effectively irreversible under cellular conditions, those are the times you should regulate, because there's no going back. In this case, maybe you want to keep a store of the phosphoenol pyruvate around for other purposes. And we'll actually see that there are some other purposes for PEP. And maybe you just need want to keep it around for that when the synthesis of ATP is going to be even more valuable. Right? You've got plenty right now. You don't need to do that last step. Start running out of ATP, do that last step, you've got some ATP right away. So why is pyruvate such a good final product? Well, in addition to the resonance stability up here that we already talked about, right? There's tautomerization down here. So we can go back and forth between a pyruvate enol form and a pyruvate keto form. The keto form ends up being a little more stable than the enol form. But if you look at the reaction, right? It was locked in the enol form to begin with, right? So it was locked in like the little more unfavorable final product state because of that phosphate. But the moment you release that phosphate, now it can relax into essentially two final products, one of which is even extra stable. So because pyruvate ends up being chemically a lower energy state than phosphoenol pyruvate, it gives you a big negative delta G despite the synthesis of new ATP. Well, that makes sense. So in our payoff phase, we oxidize glyceraldehyde 3-phosphate. That means we reduce NAD at the same time, right? Then we move that phosphate around, transferring it from the 1,3-bisphosphoglycerate to ADP, making some new ATP, right? Times 2. Times 2 for our NAD to NADH as well, right? Then we, the phosphoglycerate mutase moved our phosphate from the 3 position to the 2 position. That allowed for the dehydration of 2-phosphoglycerate into phosphoenol pyruvate, which sets us up for a great payoff. Phosphate from phosphoenol pyruvate to ADP, and pyruvate is a final production. So we got so 2 NADHs is a payoff here, 2 ATP here, 2 ATP here. Overall, we look at every step that we just talked about. We can add up all our reactants and all our products and all of those different delta Gs. And under standard conditions, the overall pathway ends up with a negative just about 30 kilojoules per mole. And you can see actually, that's just about what the last step provided, right? Without that last super stable product, you don't end up with a net negative for the entire pathway. The net production is of 2 ATP because you used 2 to start with, but you end up with 4 new ones. You also get 2 pyruvates and 2 NADHs. And we always add this plus H plus because to balance things out. So there's an extra proton released into the wild. So now the key thing is glucose is gone. That glucose is no longer available for other things glucose is valuable for. But NADH and ATP are valuable for lots of different things, and pyruvate is a great starting material for lots of different things. So the overall regulation of this process is going to look at the energy state of the cell. You have lots of ATP, slow this down. You have no ATP, you better speed it up. And how do you know you have no ATP? Well, ADP concentration should be a lot higher. Those were standard condition delta G dimes. Obviously, cells are dramatically different. I kept talking about, oh, keep concentrations of products low and reactants high and so on, like which takes you out of standard conditions already, right? So what does this look like if we look at reactions in the cell? So this was the most unfavorable step in the reaction. This is fructose 1,6-bisphosphate getting cleaved in half to produce dihydroxyacetone phosphate glyceraldehyde 3-phosphate. Under standard conditions, plus 23.8 kilojoules per mole. In the cell, weakly favorable to at equilibrium. Why?
The concentration is not at the 1 molar.
Yeah, because your reaction quotient is far from the equilibrium reaction ratio of products and reactants. The further away you are, the more energy you can get out of the system, right? The more that spring is compressed or stretched. You can actually measure the concentration of all these compounds in cells. And so this is the metabolite concentrations of various types. A lot of these you've met, right? Over the course of all the stages of glycolysis, right? You're going to end up producing different concentrations of each of these products and reactants. And none of them are close to 1 molar, right? And they're wildly different in terms of their relative concentrations in the cell. So erythrocytes, those are red blood cells. Glycolysis is incredibly important for keeping your red blood cells alive. They're one of the few cells in your body that doesn't use oxygen at all. Can anybody tell me why? Well, they don't have a nucleus. That's also part of the strategy. It makes them squishier too.
No mitochondria.
What's that?
No mitochondria.
No mitochondria yet, because if you don't have oxygen, you don't really need the mitochondria for oxidative phosphorylation. But think of it like more basic. Like zoom out. Why would you not want red blood cells to use oxygen? Yeah, exactly. Like they have a job, it's to move oxygen elsewhere. If they're using it up, like they're not very good workers. So basically, the strategy there is don't let red blood cells use the oxygen they're supposed to be moving around. So they're highly dependent upon glycolysis, and so this is even more regulated in red blood cells. Right? Okay, so different concentrations of our reactants and products. Let's find fructose 1,6-bisphosphate here. There's its concentration in millimolar. And the two products, dihydroxyacetone phosphate, glyceraldehyde 3-phosphate. We can now develop our reaction quotient here. So products over reactants. If you do this and you have different numbers of products and reactants, you better convert into molar. Otherwise, this doesn't work. Convert into molar, and you see, 0.000019 molar times 0.0014 molar. That's your concentrations here over your reactants, and you get a reaction quotient. It's a very small fraction. It's equivalent to about 1 over 11,000 or 12,000, right? If you take that ratio, right, and actually throw it into the equations we met earlier that allow you to convert a reaction quotient into a delta G, right? Here's the standard delta G, right? That's our delta G prime plus RT natural log of this ratio, and we get negative 0.35 kilojoules per mole. It's effectively at equilibrium. Right? That's barely away from equilibrium. Right, and this makes sense. This is one of those middle reactions in an overall chain where we have heavily regulated things up front, steps 1 and 3, heavily regulated thing at the very end, step 10, everything else is going to end up just kind of relaxing to equilibrium, isn't it? Because you have more or less like 3 spots for spigots, like that you can turn on and turn off, or dial up and dial down. Everything else in the middle is just going to go with the flow. And that's basically what we see. When you look at all 10 reactions, standard conditions versus cellular conditions, remarkably different. The 3 red steps, or the 3 highly regulated ones, are the ones of the big negative delta G's under standard conditions and under cellular conditions. They're the ones that are effectively irreversible in cells, because in cells, those are the ones that are going to go forward, if regulation will allow it. So you'll also notice the other big negative delta G here, step 7, that first ATP creation step, is basically at equilibrium under cellular conditions. It's in the middle. So overall, thermodynamically unfavorable reactions are kept near equilibrium. And so that means a little more reactants will just sort of push things forward. Once those deplete, right, now it's at equilibrium again. So you can add more stuff upstream, it'll flow down, and everything will kind of even out. That's basically how these enzymatic chain reactions work. The process then is driven by these 3 highly favorable reactions that are heavily regulated. And we talked about ways they're regulated. Feedback inhibition is one of the key ones, right? If you have lots of products of the overall 10 steps, don't make more. Slow it down. If you need more ATP, speed it up. And I think this is a good way to kind of visualize in a reaction coordinate diagram what this looks like, right? If you looked at just standard conditions, that's their blue line down here. This is a reaction coordinate diagram. I totally made up the barrier energies, by the way, so don't take those as some sort of real value, but just kind of big enough so that you can see the different steps, right? You start at the same place, you end lower, so it's a spontaneous reaction, right? But under standard conditions, you're kind of going up and down, and then you climb back up, and then eventually you fall, right? But under cellular conditions, you start here, and by the 3rd reaction, you're already down here. And then you stick basically at equilibrium for a while, and then you're done, right? And that's basically how it works in the cells. That you regulate this first step because it's going to be hard to go all the way back. Regulate this 3rd step because it's going to be hard to go all the way back, and you're fully committed here. But then once something flows over this mountain, it's just going to go around, right, until it falls off the last step. So this is the general strategy you'll see overall when you're considering chains of reactions, is whatever the most energetically favorable steps are, they're going to be driving things forward. The most energetically unfavorable things are going to slow everything down. The barriers, right? What is the slowest step in the reaction? Okay. So consider a metabolic pathway, 3 steps. A is eventually converted into D, going through B and C, right? Enzymes 1, 2, and 3 catalyze these steps. The reaction rates forward and backward are different. If you actually just compare these values, they're not different by much, right? The net rate forward of reaction 1 is 10 versus, reaction 2 and 3, also 10. But the ratio is completely different, right? K1 and K minus 1 are completely different. So while the net difference of this first step is only 10, this is, 1,000 times. It's 1,000 times faster, right? So the forward reaction is 1,000 times faster than the backward. This is about equal, right? Ratio-wise, these are about equal. So this is what's going to be the slowest part, the regulatory part of the entire process. This is far from equilibrium, right? Because if the forward rate of a reaction and the backward rate of a reaction are basically the same, you're basically near equilibrium. This is well out of equilibrium, so it's going to be dictating the entire kinetics of the entire chain. All right, and so where are we going to go from here? I know we're just about out of time for today. 12:10. So glycolysis is this one green box. Okay. The next stages that we're going to learn about, and basically we're going to get through the entire citric acid cycle tomorrow. We're going to take the products of glycolysis that includes those two pyruvates. Those two pyruvates can do lots of different things and go in the citric acid cycle, also known as the TCA cycle and the Krebs cycle. And you'll see more production of NADHs. So the production of NADH from glycolysis will eventually end up in the electron transport chain, which will lead to oxidative phosphorylation and the production of a lot more ATP. So glycolysis happens in cells without mitochondria, like eukaryotic cells and prokaryotic cells. If there are mitochondria available, the products of glycolysis go to those mitochondria and can allow you to extract a lot more energy than ATP allows, right? We took one glucose and made 2 ATP on net. Not a big turnout. All right. So pyruvates has lots of potential products that it will create. There's still a lot of energy left in pyruvate. It's more oxidized than glucose was, right? But it can be oxidized all the way to CO2. And that's the role of the citric acid cycle, is to oxidize pyruvate all the way to CO2. Instead, if you don't do that, pyruvate can go through these other processes. Under hypoxic or anaerobic conditions, pyruvate can be fermented. Some species will ferment to ethanol, some will ferment to lactate. Okay. Under aerobic conditions, that's when you get the pyruvate going into this TCA cycle. I think I'll leave it there because I threw a lot at you today, the entire glycolytic pathway, right? So I'll let that sink in, and we will cover fermentation and the citric acid cycle tomorrow. The citric acid cycle is presented a lot like the glycolysis cycle, with each of the steps, all the details, all the justifications, and so on. So please read ahead if you can, just kind of like absorb that material. All right. All right. I'll see you later. You have a question?
Yeah. So this is like super random and kind of looking ahead. And that we put a metabolic map like on our homework. Should we like commit that to memory or?
No,
Okay.
but it's always useful to provide context,
Okay.
right? Like, because we're looking at one little thing, and it's just all connected, and the regulation of these things doesn't come from like nowhere.
All right. I just wanted to make sure.
It's
I don't know. I feel like it's better to ask.
Yeah, well, I think it's useful just to kind of keep in mind that like there are other ways that things enter these cycles and they exit these cycles, and we'll talk about some of those, but not nearly all of them.
Okay.
And plus, you all want to be doctors anyways. So you're gonna need to know this stuff.
I don't understand that.